So soybean meal contains significant amount of carbohydrates, and a large proportion of these are GOS. So these molecules occupy space in the diet, but don't contribute usable energy unless they are broken down. So that's why the importance of adding alpha-galactosidase. So these oligosaccharides are hydrolyzed before they reach the hindgut, breaking them into simple sugars that animals can actually absorb, and therefore improves nutrient availability, supports gut comfort, and boosts overall performance. So in short, an alpha-galactosidase converts what would otherwise be an anti-nutrient into a source of useful energy, making soybean-based diets far more efficient.
Hello, everyone. Welcome to another episode of the Poultry Nutrition Blackbelt Podcast, where we discuss the latest in poultry nutrition research and industry trends in approximately 10 minutes. My name is Sam Rochel. I'm a co-host of the show and associate professor of poultry nutrition here at Auburn University, and joined today by Laura Guarás, who is a research and development manager at Kerry, who's with a background in applied enzymology and is going to talk to us today about alpha-galactosidases and what that can mean in poultry nutrition or animal nutrition. So thanks for joining, Laura, and great to talk with you.
Thank you, Sam. Nice to meet you and nice to be in this podcast today.
Yeah, very good. So just to get us started off, as a general background, what is an alpha-galactosidase and why is it relevant for animal nutrition?
Very good question to start with. So an alpha-galactosidase is an enzyme that targets a specific group of carbohydrates called galactooligosaccharides, or in short, GOS. These are naturally found in legumes, especially in soybeans. And if you ever come across with names like raffinose, stachyose, verbascose, or augose, those are the main GOS molecules that we are dealing with. So these GOS, they are built on a sucrose core with a galactose unit attached, which gives them energy potential. But unfortunately, monogastric animals can tap into the energy on their own. And this is because poultry do not produce alpha-galactosidase endogenously. Without the enzyme, these GOS molecules present in soybean meal, for example, pass through the small intestine, undigested, and end up in the hindgut. So once they are there, they undergo fermentation, which can draw water into the gut, produce gas, and contribute to water litter and gut discomfort. So yeah, there is also a nutritional angle. So soybean meal contains significant amount of carbohydrates, and a large proportion of these are GOS. So these molecules occupy space in the diet, but don't contribute usable energy unless they are broken down. So that's why the importance of adding alpha-galactosidase. So these oligosaccharides are hydrolyzed before they reach the hindgut, breaking them into simple sugars that animals can actually absorb, and therefore improves nutrient availability, supports gut comfort, and boosts overall performance. So in short, an alpha-galactosidase converts what would otherwise be an anti-nutrient into a source of useful energy, making soybean-based diets far more efficient.
Proven on the farm, trusted on the plate, let our technologies help make your production goals a reality. Learn from the experts how carbohydrates can improve nutrient utilization, gut health technologies differ by type, and how liquid smoke can light your bird performance on fire. Kerry isn't just leading in animal agriculture, we're innovating it.
Very good. And we know, a lot of people forget that when we look at the gross energy between corn and soybean meal, corn is our main energy source. The gross energy is not that different between those. But then when we look at the ME value, we see a big penalty on the soybean meal. We know that the carbohydrates that you're mentioning are a big part of that. So, you touched on this a little bit, but really what value does an alpha-galactosidase bring, and why is this particularly important? We look at soybean meal prices today, maybe hovering $350, whereas a couple years ago, $450 per ton. So we see a lot more opportunity with soybean meal, maybe, and these enzymes to really add value.
Yeah, but one of the primary benefits is unlocking more energy from soybean meal, as I say, as in soy inclusion rises in poultry. So that's the amount of this GOS entering the gut. So without the enzyme, without the alpha-galactosidase, the energy remains trapped in undigestible GOS. So with the enzyme, those GOS are transformed into something that the animal can actually use. And this leads into several practical outcomes. So better nutrient digestibility, improve gut condition, more predictable feed conversion, and more efficient use of every kilogram of soybean meal. And why is this especially important today? The answer lies in market dynamics. Soybean prices fluctuate constantly, and when prices drop, like currently, currently the price of soya is quite low, so soy becomes a more attractive ingredient. So nutritionists tend to increase soy inclusion to optimize cost. But this also means higher GOS levels in the diet and a greater need for alpha-galactosidase to keep performance steady. Yeah, and there's also one factor is the variability in the concentration of GOS, and in soybean meal, which depends on season, climate, geography, quality of the crops, etc. So this variation can be significant, and that's why also an option could be superdosing the diets with alpha-galactosidase, higher inclusion levels that help ensuring consistent breakdown of the GOS, even when the raw materials vary.
Sure. Yeah. And so you mentioned the substrate, basically these different oligosaccharides, different sizes. And can you talk more about kind of the amount and the substrate a bit within soybean meal? And are there any potential indirect effects beyond, the direct energy release from the substrate?
Yeah, so while alpha-galactosidase plays a critical role in targeting raffinose family oligosaccharides, we know that multi-enzyme strategy is essential for achieving full non-starch polysaccharide degradation in modern monogastric diets. And this is because the non-starch polysaccharides such as arabinose islands, beta-glucan, cellulose, among others, form complex and highly branched architectures that cannot be effectively dismantled by a single enzyme. So combining enzymes such as cellulases, beta-glucanases, silanases, and other debranching enzymes work synergistically with the alpha-galactosidase to one could be like attacking different bonds within the NSP matrix, breaking down both the linear backbone and the branched structures. And doing that, you improve the accessibility for complementary enzymes, and the outcome also will be maximizing release of trapped nutrients and energy. So we believe that multi-carbohydrate solution therefore delivers a complete and extensive coverage for hydrolysis and utilization of non-digestible fractions of polysaccharides. And this is a comprehensive mode of action that ensures more efficient degradation of NSPs and better gut function, nutrient absorption, and overall performance in the animal.
Yeah, that makes perfect sense. And so as you mentioned, the mode of action of the multi-carbohydrates and the alpha-galactosidase, are all alpha-galactosidases the same? Or are there different types of enzymes and how they're applied and ultimately their performance?
So yeah, as I was saying, the alpha-galactosidase, they come from different microbial and plant sources, and those origins create natural variation on how the enzymes behave. So differences appear in, like, for example, substrate specificity, which cause structures they hydrolyze most effectively. The stability is how they tolerate heat, pH changes, gut conditions, and structure, which influences how they bind and break down the substrates. So what we see in our research is that we often see an exceptionally strong performance from legume-derived alpha-galactosidase, which are highly effective in breaking down legume origin GOS. So almost like they are naturally matched. It's like a nice example of like nature working on nature.
Yeah, sure.
Yeah. And another important factor is how substrate profiles vary. As I mentioned before, like host composition is influenced by different factors. So if the substrate itself varies, we need enzymes that are robust enough to handle these swings, and also superdosing to counteract this variability. And that's why even within Kerry, we do enzyme developers like we do spend a lot of time studying ingredient profiles, building data sets, and testing across different diet matrices. And understanding this variation helps inform enzyme design so that performance stays consistent regardless of the diet or the season. Yeah.
Sure. Yeah. No, it makes sense. And as you mentioned, these are naturally present enzymes from a lot of different organisms. But obviously, when we talk about commercial scale, bioengineering is key to be able to get the yield and the quantities and improve strength. So, how is that? How are advances in this field, shaping the alpha-galactosidase solutions from Kerry?
We're in a crazy time because bioengineering is opening up a new era for feed enzymes. With advances in protein engineering, strain selection, and fermentation technology, we can now design enzymes that are more specific to the target substrates, more efficient at hydrolyzing GOS, but also other substrates. They are more stable during pelleting and storage, and they are better adapted to gut conditions across different species. So what it does, these improvements mean that the enzyme can survive harsher processing conditions, activate earlier in digestion, and work more efficiently in vivo.
Okay, yeah.
But the innovation doesn't stop in the lab. We need real world validation. And every new engineering enzyme must be backed by animal trials to show the benefits actually translate into improvements.
Yeah, absolutely.
And just the last point is like, from development perspective, we are really, really beginning to tap into what is possible in this bioengineering, biotechnology scene. So there are many tools to advance, and we'll see enzymes that are even more targeted, more resilient, and more integrated into precision nutrition strategies. From a Kerry standpoint, we are deeply, deeply committed to advancing this frontier, combining biotechnology and real-world testing to build the next generation of enzyme solutions for animal nutrition.
Absolutely. Well, I appreciate that perspective, especially as someone who's on the side of the application and looking at how this works in the animal. It's always nice to get insights from someone who's an expert on the actual enzymology side and R&D and how these are developed. So really, really appreciate your insight and perspective on this stuff.
Thank you. Thank you for your time too.
Thank you. Well, really enjoyed the conversation, and thanks to all of you for joining another episode of the Poultry Nutrition Blackbelt Podcast. If you enjoyed this, please like and subscribe on your favorite platform, and we'll catch you on the next one. Thank you.
Mm-hmm.